Every organism has to have energy and nutrients in order to survive, in addition to water, of course. So we're going to take a look at those topics in this lecture. Organisms can be classified by what we call trophic levels. These are feeding levels in the food chain or food web. Now, autotrophs are organisms that are able to produce their own food, so to speak. They use inorganic sources of carbon, and then they use energy. Those that are plants and are capable of photosynthesis use light energy to do this. And then we have some organisms that are chemosynthetic, and they use inorganic molecules as sources of carbon and energy. They're not, light is not available to them. So autotrophs are literally self-feeders. And then all of the other organisms are heterotrophs, or other feeders, and they will get their food from an autotroph or another heterotroph, as we would see in a food chain. Let's talk about solar energy here. The world is solar powered. Light propagates through space as a wave and as a particle. I know that's kind of unusual, but the physicists use those terms. Now, a particle is a packet of light energy, and the wave is, of course, moving through the environment like that in a pattern. And so we have the different wavelengths have different colors. The light that is important for photosynthesis is the PAR, and it's in the visible range, as we will see. Infrared is not used, and ultraviolet is not used. Those are outside of this PAR, and we can't use those for photosynthesis. This PAR strikes the Earth, and notice we get a large amount of it reflected, and we get absorption. So this is our boreal forest example. So in this case, not much of this makes it to the ground, so these guys down here have to be adapted to low light. And you can see most of this absorbed by the plants in the upper levels and in the higher levels, and then we get reflection from there. So only the light that is absorbed is going to be useful for photosynthesis. Now, PAR can be quantified, and the physicists do this, at photon flux density. And it's really just the number of photons striking a square meter of surface per second. And chlorophyll absorbs light as photons. Now, landscapes, water, organisms can all change the amount and quality of light that might be reaching a particular square meter of ground. The photosynthetic pathways, we have two ways that plants go about this photosynthesis. One is called the C3 system, and we'll see why that is important. The other one is called the C4. Now, the C3 is used by most plants and algae, and what happens here is the carbon dioxide combines with ribulose biphosphate to give a 5-carbon sugar, and then we get this phosphoglyceric acid, which has 3 carbons as a result here. Now, in order to get this done, the plants must open their stomata to let in CO2, and the water may escape because of the gradient there through the stomata in transpiration or evaporation. So here's a C3 photosynthesis, the typical type of photosynthesis. So here's a C3 plant. Here's the structure of its leaf. We can see the little holes here that are on the bottom side, the stoma or stomata. So there's one, so we can get gas exchange through that hole. Notice the structure: epidermal cells, muscle cells, bundle sheath cells, and then the veins of the plant. So here's what happens during C3 photosynthesis. So we make this PGA, which is a 3-carbon acid, which is why we call it C3 photosynthesis. Notice this occurs in the mesophyll cells, and then we get the final product. So this PGA, we can turn it into sugar, starch, and other types of molecules. C4 photosynthesis is important in some environments, particularly those that are dry. So this helps the organism to conserve water. So they don't need as many stomata to be open. And the acids produced in this carbon fixation diffuse into specialized cells that surround that bundle sheath. So here's a plant, corn plant here. A lot of weeds are also C4 plants. So notice we have the epidermis, the mesophyll, and the bundle sheath cell. Here's the vein. Here's the stoma. Now this is more complicated. So the C4 plants take in CO2 and they combine it with PEP, which is going to form a C4 acid. Notice this is occurring in the mesophyll cell, and then that C4 acid is moved over into the bundle sheath cell, where we can then follow the regular pathway to make sugars and starches. Notice the pyruvate cycles back to make PEP again. So the PEP acts as kind of a shuttle in order to move that C4 acid into the bundle sheath cell. So the action takes place in the bundle sheath cell, and these organisms can survive in drier climates. Then we have another type of photosynthesis, CAM photosynthesis, crassulucian acid metabolism, and this is limited for succulent plants. So maybe something like your aloe vera that you keep at home. These plants are found in arid or semi-arid environments, and the carbon fixation actually takes place at night. That reduces the water loss. Now they have low rates of photosynthesis, but extremely high rates of water efficiency. So here's CAM. So CAM plants open their stomata and take in the CO2 at night. Humidity's higher, temperature's lower, so you won't lose as much water to evapotranspiration. Now here during the day, the CAM plants close their stomata when they would be losing water, so they're preventing that loss. Notice their leaves are, in this case, just thorns. So here we go in the mesophyll day at night, and then in the mesophyll in the day, we're following the usual pathway to produce our PGA and the sugars and starches and other materials that are needed by the plant. Now photosynthesis is much more complicated than this, but this understanding will take care of our needs in this course. Okay, heterotrophs. 3 feeding mechanisms we have. We have herbivores, carnivores, and detritivores. So herbivores quite simply feed on plants in the terrestrial environment, or they may feed on algae in the aquatic environments. Carnivores eat other animals, and detritivores feed on non-living organic matter, so all that stuff that accumulates at the bottom, say, of a forest floor, they'll eat that. It's not alive, but it contains organic matter. Now, 5 elements make up most of the biomass of any type of organism, and these are carbon, hydrogen, oxygen, and nitrogen. And I like to remember that little acronym there. Okay, that's probably about 95%. And if you add sulfur and phosphorus, then you've got about 99%. So carbon, hydrogen, oxygen, nitrogen, sulfur, and phosphorus make up most of the organism. That goes for you too. Other nutrients that are essential for plants, and a lot of these for us also, but you can see this list. So these might be what we call trace elements that are required in much smaller amounts. So we have some mostly metals here, don't we? And then some other minerals. Herbivores can have some problems. For one thing, the food that they eat has low amounts of nitrogen, and so they may have to eat a lot of it. They must also overcome the plant's defenses. The plants have physical defenses. They produce chemicals. They produce indigestible material, cellulose, lignin, cilia, toxins, compounds that reduce the digestion of the organism. So these herbivores have a big problem to overcome. So detritivores consume food that's rich in carbon and energy, but it's, again, poor in nitrogens. Like a dead leaf may have only half of the nitrogen content of a living leaf. Now, fresh detritus may still have some chemical defenses present, so that may prohibit the organism from utilizing those as food. Carnivores, on the other hand, consume prey that's nutritionally rich. They've got some problems, though. The prey is trying to escape them, so they have defenses. They may have warning colors, and they may use mimicry. So sometimes an organism that tastes bad will mimic an organism that doesn't taste, or vice versa. An organism that doesn't taste bad will mimic an organism that tastes bad, and so gain protection from that. The monarch butterfly and the viceroy butterfly are good examples of that. And I can't remember which one tastes bad, but I suspect it's the monarch because they feed on milkweed. So that would be Mullerian mimicry. Well, actually, that would be Batesian mimicry, and the warning colors would be Mullerian mimicry. So these guys are saying, "Don't eat me. I'm not good for you. I may be poisonous." So here's an example of Mullerian mimicry amongst the butterflies. So these guys resemble each other, but not all of them are noxious. Some of them will be just fine. In the case of birds, sometimes they have to learn by actually taking a bite of a nasty tasting one. They spit it out, and that means they won't feed on any of these guys because they learned that those don't taste very good. It might take the bird a time or two to learn that. Yeah, the monarch, I was right. The monarch is loaded with toxic substances, some cardiac glycosides, and it gets these from the milkweed plants when it's eating as a caterpillar, and these render it unpalatable to many of its predators. And so here is the viceroy, and here is the monarch. So we've got the mimicry here. And I'll put this link in the in Blackboard. Predators are usually a selection agency for the refined prey defense. So we really have co-evolution here. We kind of have an arms race going on, and the predator and prey species are evolving to kind of try to match the activities of the other. So we call this co-evolution. Inorganic molecules can also be shown to have chemosynthesis. So we have in 1977, they discovered these deep ocean vents and these tube worms living in these deep ocean vents. Now they utilize, actually they contain bacteria that utilize the materials being discharged from the oceanic substrate through hot vents. So we have autotrophic bacteria. So here's another example of those. So the Earth's hot, the seawater filters in, and it heats up, dissolves materials out of the rocks, and then we have the material come up. We have black smokers and white smokers. These are white smokers. And the seashore is, in some cases, spreading at this point in these different directions. So here's the black smokers, hot springs of the deep sea, the Juan de Fuca Ridge, and notice 14 stories tall, pretty tall structure, and we've got unique life forms here. Now they get their energy from these sulfates, and the bacteria feed the sulfates. The bacteria use the sulfates, and it's totally black down here. And you'll want to take a look at that link in order to see the story there. Energy limitations. Now there are limits on the potential rate of energy intake by organisms, and they've studied the feeding rate and food availability here. And particularly plants have been studied by studying the photosynthetic, can't talk today, photosynthetic rate to the photon flux density. So the rate of photosynthesis increases with flux density at low light levels. It rises more slowly at intermediate light intensities, and then it tends to level off at high light intensities. So there can actually be too much light, too many photons striking the leaf, and that will actually inhibit photosynthesis. So notice we have that photosynthetic rate on the y-axis, flux density on the x-axis, and see how it levels off. So in other words, it can only handle this number of photons, the photosynthetic systems. Food density and is important here. So Holling described three functional responses. The first one, feeding rate increases linearly as food density increases, levels off at a maximum. Feeding rate rises in proportion to food density. And then the feed rate increases most rapidly at intermediate densities. So you get the S-shaped curve, and here are the three curves that we can see, type 1, type 2, and type 3. So this is the S-shaped curve he was talking about. Optimal foraging theory assures that if energy supplies are limited, then the organisms cannot simultaneously maximize all life functions. So they must make a compromise between competing demands for resources. So this is allocation. So there may not be enough food to reproduce, but there might be enough food to stay alive, for example. So if everything's equal, more abundant prey yields larger energy return, but you've got to take into account the energy expended searching for the prey. And you've also got handling time when you're trying to kill the prey. And so the organisms tend to maximize the rate of energy intake. I was watching a video of cheetahs feeding in the African environment, and the cheetah can only hold maximum speed for about 10 seconds. So if it doesn't catch its prey during that time, it tends to stop in order to conserve energy because it wouldn't be worth it to continue the chase. So this is the bluegill sunfish. And notice the most abundant prey are about 1 millimeter long, but the optimum foraging theory predicts that the bluegill should feed on prey 4 millimeters or longer. And as the prediction followed the research, that's what they did. Most of their food items are 4 millimeters in length. So they're picking the right size. If you're picking these small guys, you would be expending a lot of energy for not very much return. So limited sources of energy is going to be a problem for plants too. So how are they going to allocate that to the leaves, stems, and roots? Now a researcher named Bloom suggested that plants adjust the allocation in such a way that all resources are equally limited, and they appear to allocate growth in a manner that increases the acquisition of resources when the resources are in shortest supply. So they may make more leaves, or they may make more roots as necessary in order to fulfill their needs. And there's a moray eel, a predator in the ocean. So that guy can give you a really good bite. And that's all for this one.